Jim, for giving us the opportunity to present our work here on this wonderful workshop today. So as you can see from all these logos, we're quite an interdisciplinary group. So my name is Fostier. I'm from Ghent University, but by all means, most of the work that I will present here today was done by the principal author, which is Dries de Kap, but he is currently on a 1-month holiday to Taiwan, so he couldn't be here. Other people are Joke Reumers from Janssen Pharmaceutica, which is the pharmaceutics division of Johnson & Johnson. Charlotte Herzeel, who is working at IMEC, and then Pascal Costanza, who is working at Intel Belgium. And all of us have in common that we are working in this lab, this ExaScience Life Lab, which is located also in Leuven in Belgium. And this lab has the goal to, let's say, advance computational life science through high-performance computing. And one of the subjects is the acceleration of sequencing pipelines, and that's the talk for today. So it's called Halvade, Scalable Sequence Analysis with MapReduce. So most of you know that the human genome has been around for quite a few years now. So in 2001, there were, well, it was considered ready, right? There were two efforts, a public and a private effort. Papers came out at the same time. That was in 2001. Well, in 2003, it was officially declared finished. We're now at revision 19, just a few months ago. And back in those days, this effort cost about $3 billion US, so that's roughly about $1 per nucleotide. So when we want to sequence another individual, we can rely on this reference genome, this individual or number of individuals that were sequenced a few years ago, simply because of the fact that they are 99.9% identical. And this will help us because we only need to figure out which are the 0.1% differences between the reference genome that we have and the patient for which we want to establish the genome, right? So this is a two-step procedure. Many of you are familiar with this. Patient comes in a hospital, for instance, delivers a sample with DNA, for instance, blood, and this is being sequenced by a sequencer, and 90% of the sequencing nowadays is being done by Illumina, mostly for resequencing purposes. And what comes out of these machines is an enormous amount of short reads. So that's the wet lab step. The second step is the computational step. You have your reference genome from, let's say, the human beings that were sequenced 15 years ago, and you have the short reads from your patients. Well, the first step is aligning these short reads to the reference genome. This is, well, this seems simple, but it is not simple. Why is it not simple? Because I've said these reads are 99.9% identical to the reference genome. So you cannot rely on exact string matching. You need to have some form of imprecise string matching, but also the reads that you have here contain a lot of errors. So these Illumina machines, they make errors. Well, not a lot of them, but they do make errors, and these errors are not random. So they are systematic, they are biased, and it takes quite a lot of domain knowledge to know how to deal with these errors. There are certain tools that do this, apparently over 100 or even more, but I think that I can safely say that BWA is one of the most important tools for doing exactly this, namely aligning reads to a reference genome, especially for DNA. So most of the people are actually using BWA. The second sub-step of the computational step is then comparing these aligned reads to the reference genome. So basically, this is just comparing character by character, what are the differences between the aligned reads, the contents in the aligned reads, and the reference genome. So for instance, there is a T here, and in all of the aligned reads, there is an A. It again seems simple, but it is not simple. There are, again, tools that contain a lot of domain knowledge that do this. One of the tools that is widely used is GATK, the Genome Analysis Toolkit by the Broad Institute. Why is it not easy? Well, again, because these reads contain errors, because these reads might be misaligned. like completely misaligned, or just a small portion of this read that is misaligned, like one small indel, and also because human beings are diploid, because they not only have, for instance, an A here, but on one chromosome they could have an A, on another chromosome they could have a T. So all of these things together makes this, again, a very challenging task, right? But in the end, you get your sequenced genome. So GATK has recently, 2 years ago, well, Geraldine van der Weera from the Broad, at least, has written a paper in Current Protocols in Bioinformatics describing a recipe called, well, the best practices recommendations to how this computational step should be done. So basically what you get here as an input is your paired end reads. These are aligned using BWA. You get your aligned reads. They get sorted. Your aligned reads, which are sorted, need to be further pre-processed. You need to have duplicate marking, other pre-processing steps, then a number of GATK steps like a local realignment, BQSR, and then finally variant calling. So all of these steps have been written out in this paper. It's very easy to read. So it's basically like a recipe. You run this tool with these parameters, then you run this tool with these parameters, and so on. And they also explain why you need to do this. And it's actually quite instructive. So this is actually our problem at hand. This is the pipeline on which we are focused, right? So this is the pipeline in more detail. So the alignment step can be done using BWA align or BWA mem. Mem is then used for longer reads. Both of them are written by Heng Li. Then the pre-processing step, the data pre-processing step, involves some Picard and or SemTools steps, where the records that have been aligned are sorted, converted to BAM format. Read group information is added if it hasn't been added before. Duplicates are marked, unmapped alignments are removed, and so on. And all of this such that the resulting BAM file is in a specific format that GATK can handle it, and that can do quality variant calling on it. So this also involves a few steps like local indel realignment, like correcting small alignment errors due to indels. Base quality score recalibration is kind of a recalibration of the per nucleotides quality score that comes along with your read to correct for certain biases in your technology. And then finally variant calling, for which you also have certain options available. Well, this graph has been shown multiple times. The problem is that, well, since, let's say, 2008 and 2014, the decrease in sequencing has, well, let's say, outpaced the decrease in compute cost by a factor of 1,000, if not more. So what does it mean in practice? Let's take a data set of a single human being. It's exactly the same data set as Frank talked about. So it's the NA12878 or something from Illumina Platinum Genomes. So what you get is 1.5 billion reads of length 100 nucleotides, right? So it's a 50-fold coverage of the human genome, and uncompressed, this is about 300 gigs of raw data. Well, if you apply on a single core this best practices guideline or this best practices pipeline that I've talked about before in previous slides, well, it takes about 172 hours to do the alignment, another 35 hours to do the data preparation, and then finally another 80 hours to do the GATK steps, including variant calling. So all of this together is about 12 days. So that's on a single core, but on a modern machine. So suppose a patient comes in a hospital, he delivers a sample, it gets sequenced, it takes about 2 days. And then it takes about another 12 days to analyze the variants or to get the variants from this sample. So our question is now, can we reduce this analysis time, significantly reduce this analysis time without having to re-implement or otherwise change the tools, BWA, Picard, SemTools, and GATK that we are using by using parallel computing? And the answer is yes, we can. Why? Because this is, again, the same best practices pipeline. So this is the alignment step. This step is parallel by read. Basically, the alignment of one read on one compute node can happen independently from the alignment of another read on another compute node. Sorting is something that has to be done, but we can do this in parallel. And then all of this pre-processing steps and variant calling steps, they are parallel by genomic regions. Once your reads have been aligned, basically the work that has to be done on chromosome 1 can happen in parallel with the work that has to be done on chromosome 2. Right? So basically something that is parallel with a sorting step in between. It's kind of reminiscent of a certain programming model, which is the MapReduce programming model. And that's the model that we use. So MapReduce basically has, let's say, big data as input. This data is being split into smaller chunks that have to be independently processed by a number of mappers. This is done in parallel. These mappers emit some intermediate data, which is organized as key value pairs. These keys are sorted and grouped according to keys, and things that have the same key, for instance, this is a simple word counter, the word DNA, they're all being sent to the same reducer, which is a second step, a second parallel step in which certain intermediate results are aggregated. This is exactly what we do in Halvade. So basically what you get out of a sequencer is, in case of paired end reads, typically 2 Fostier files. What we do is we have a data pre-processing step in which these 2 Fostier files are being copied onto the machine where they are being processed. Basically what we do is we chop these files into smaller chunks. We also interleave those files such that we have paired end reads stored adjacently, one next to the other, right? This is important for data locality during the alignment step. The mapping phase in MapReduce corresponds to alignment, right, where we have parallel instances of BWA, each operating on a chunk of the input data. The data pre-processing and variant calling happens in parallel during the reduce phase. In between map and reduce phase, we sort all the aligned SAM records according to position. So basically our key consists of 3 values. One is a chromosome number, the other one is the position within the chromosome, and then the third field is, let's say, a chromosome segment. So basically what we do is we take the complete human genome, we chop it into an arbitrary number of approximately equally large chunks, and these define the reduce task, right? So in our case, a reduce task is about doing the variant calling on a piece of a chromosome of about the size of 2.5 million nucleotides, right? So all of this is encoded here, and the value is then, of course, simply the SAM record. The shuffling ensures that all of the aligned reads that are required by a certain genomic chunk are being sent to a specific reducer. They are then further sorted according to aligned position, and then in the reduce phase, variant calling and stuff happens. Basically, we get a VCF file out of each reducer, right, which can then be further aggregated into a single VCF file using a second MapReduce task. Because these VCF files are really small compared to these Fostier files, this second MapReduce task is really lightweight, so really fast. So in more detail, we have our 2 Fostier files. They are being interleaved and they are being chopped into chunks which are smaller than 64 megabytes because this is the default block size within an HDFS, and we want to ensure that one chunk is being placed into a single HDFS block, so on a single node, so perhaps it's duplicated 3 times on other nodes as well. That's not really important. What is important is that we have one chunk in one HDFS block, right? So this is some remote reading and writing while we copy our stuff onto the HDFS. But then when we launch the MapReduce job, all of the BWA instances will have local reading from their own disk. So basically we are processing the data on the nodes on which they are stored, or at least the MapReduce programming framework ensures that this is done. And because a certain chunk, for instance, this chunk 1 is being replicated on several compute nodes, there is quite some flexibility to ensure that this can really happen. So this is the BWA step. So key value pairs are being emitted here. These key value pairs are being shuffled to the right reduce to the right reducer, and then secondary sorting is used to further sort them according to aligned position. So what you have at this stage is that you have your complete genome, right, from region 1 up until region M. You can define how many regions there are, and all of the reads that align to that certain chromosomal region are also being sent to the correct node, right? So in case you have a certain chromosomal region, for instance here, region 3 and 4, for which there are reads that span both positions, right? So these reads are being sent to region 3, these reads are being sent to region 4. In case you have reads that span the interface between adjacent regions, they are being sent to both, right? To ensure that all of the read information is available for GATK to do accurate variant calling near the interfaces. So then the reduce stage where you have your genomic regions, which are being processed in parallel by Picard and by GATK, perhaps also SAMtools. We use SAMtools for certain steps because SAMtools is multi-threaded and Picard is not. All of these data preprocessing things can be replaced by LPrep. So LPrep is a single stage drop-in replacement for Picard and SAMtools that does all of the sorting, all of the cleaning, all of the duplicate marking, and so on, but it's a lot faster than SAMtools and Picard. And because we make no modifications to BWA and GATK, we support different versions. In order to have good performance, two things are important. First of all, that's a task size. If we have very little, very few, very big tasks, we will have problems with load balancing. If we have a lot of, let's say, meaningless tasks, we will have a lot of overhead scheduling those tasks, tool initialization overhead, and so on. This is indicated here, where we use, for instance, for the map phase, where we use input chunks of 240 megabytes. That's too big. You do not have a good load balance. This is slightly below 64 megabytes. So remember to have just one chunk per HDFS block. You have good load balance. Second thing that is important for optimal performance, that's the number of mappers and reducers that you're going to run per node. Basically, if you have nodes that contain, I don't know, 24 CPU cores or 32 CPU cores, and you're going to run a single instance of GATK, you're not going to get a very good speed up compared to single core performance. This is because the multi-threading within GATK, but not only GATK, Picard, for instance, is even worse because it is single-threaded, it is not multi-threaded. But the multi-threaded scaling becomes, let's say, well, not so good, especially when you move to a large number of CPU cores. So basically, it's far more efficient to have several instances of the same tool run in parallel on different chunks of data. So what we do is instead of running one task per node, which takes about, I don't know, 1,000 seconds. If you have 2 tasks per node, so this is 24 cores per task, this is 12 cores per task, you see that the runtime approximately halves. This is 400 when we have 4 tasks per node, and when we move to 8 tasks per node, we get other problems. So in this case, it's basically because we do not have sufficient memory for all of these instances to run in parallel. So in our case, 4 tasks per node is optimal. So I was talking about the multi-threaded scaling behavior of GATK. So these are a number of different modules within GATK that are relevant for our pipeline. As you can see, in the best case, the unified genotyper has, let's say, modest scalability, but let's say this print read module is performing well. Let's say it's slower than single-threaded behavior. So it's not so good. So the overall performance, multi-threading performance of GATK is not so good. So I was talking about LPrep as a drop-in replacement for the data preparation. This is a benchmark of LPrep. So this is the standard combination of SAMtools and Picard, and you can see that the different colors refer to different things. This is Sambamba, which is already a better implementation, and this is then LPrep. And basically most of the speed gain comes from the fact that all of these tools have different stages in which a lot of data is being read and written to disk, and LPrep does all of this in memory. But LPrep requires more memory than the other tools. Some benchmark results. So the first thing where we benchmarked was on an Intel big data cluster in Swindon. So this cluster has 15 nodes. Each node has 24 CPU cores. I think it's Ivy Bridge, so it's fairly modern CPUs. Every node has 64 gigs of memory. You have a pretty fast interconnection network, so it's not a high-performance computing InfiniBand network, but nevertheless it is a fast interconnection. So in total, what we have is 360 physical cores. So when we use hyperthreading, that's 720 threads that we run in total. We have about 1 terabyte of RAM. And this cluster, well, because it's an Intel cluster, it runs on Cloudera 5. So as mentioned before, we use 4 tasks per node. So on each node, when we do alignment, we're going to run 4 BWA instances in parallel, or 4 mappers in parallel, and the same for the reducers. So this is the speed up, the multi-node speed up that we get out of this, which is basically, pretty good. So one curve is the speed up, the other curve is the efficiency. Basically, using 360 CPU cores, we get a parallel efficiency of over 90%, which is quite good. So this can also be seen from this resource occupation graph. So the red part is the map task, and you can see that there is almost perfect occupation of all the nodes. You can see a slight load misbalance, but not a lot of time is being lost here. Shuffling and sorting is being done during the reducing. So you can see that it is not a big fraction of the time that is being spent during reducing. So most of it is being spent during Picard and GATK. And we also have a fairly good load balance, right? We also tested the implementation on Amazon. So we moved to the C3.xx large nodes, which are more or less the same as the as from the Intel big data cluster. So few more, a little less or fewer cores per node, 60 gigs per node. Enhanced network performance, which is also probably 10 gigabit per second. In total, we have 256 physical cores, about the same amount of RAM, and we again run 4 tasks per node. So we get a very similar speed up curve and parallel efficiency curve on Amazon. So it's also about 90% of efficiency. So what does this mean in real numbers? So for instance, on the Intel big data cluster, using a single node, we get a runtime of about, what is this, 2 days, right? Which is already, a lot less than, running this pipeline in parallel using only multi-threading, because then you would get 5 days, right? So this speed up is solely due to the fact that in this case we have 3 instances of GATK running in parallel using only 1/3 of the available cores, instead of having 1 instance of GATK using all of the cores. When we move to 15 nodes to all of our parallel tasks, this comes down to a runtime of 2 hours and approximately 40 minutes, and this is more or less the same on the Amazon cluster, right? So under 3 hours. We spent quite a lot of time figuring out, so when we were analyzing variants at the end, so things that we got at the end, the variants, so we were wondering, are they exactly the same as running the sequential pipeline? And the answer is no, they are not exactly the same. And we figured out why this was. Well, first of all, they're not exactly the same, but if there are differences, so this, for instance, this blue line are variants that were only found in the sequential run of the pipeline, and then the red line are variants that were only found with the parallel Halvade run of the pipeline. Well, all of them, or nearly all of them, corresponds to variants with very low variance confidence score, right? So basically, variants that were called because of one supporting read. And And we tried to figure out what was the underlying cause of that. Basically, if you do something in parallel, you want it to be exactly the same as doing something sequentially. Well, it turns out that the majority of the variation comes from the fact that instead of running one instance of BWA over the entire FastQ file, we are running several instances in parallel over a chunk of the FastQ file. BWA is not provided with the insert length. It learns this from the input file itself by mapping a number of reads. Well, if you train BWA on the entire FastQ file, it gets a slightly different insert size than training BWA from, let's say, a middle chunk somewhere. And this very small difference in training can result in a read that is slightly aligned otherwise in highly repetitive regions, meaning that these variants are being called, or that a certain variant is being called in the parallel run and not in the sequential run, and vice versa. But as you can see, I think that most people use a threshold of variant confidence score 30. There is perfect concordance between the sequential run and the parallel Halvade run. So to wrap up, so if we have this pipeline and we run it single-threaded on one node, we get a runtime of about 12 days. If we use multi-threading on the same node, we get a runtime of about 5 days. So this node has 24 CPU cores, so that's not very good scaling behavior. If we use the same node with Halvade, the runtime decreases from 5 days to 2 days, so that's already a speed up of 2.5. And if we then move from one node to 15 nodes, it is below 3 hours, and we have very high parallel efficiency, right? So we are doing the same implementation. So we also support exome sequencing using BET files. So all the partitioning of the genome works with BET files. So naturally, we also support exome sequencing. And while it's no longer work in progress because it has finished, there is also a best practices pipeline for the RNA-seq variant calling, which has now also been implemented in Halvade. And we are trying to run some samples with that. So let me again acknowledge all of my co-authors. Halvade is publicly available. The source code is available, can be downloaded from GitHub. And if there are any questions, I will be happy to address them.
It's like a driver for MapReduce pipelines. Does it, for instance, automatically determine the optimal number of threads per node, or
yes, it has support for that.
Of the data, or
yeah.
And
so
it's all running in Hadoop, right?
It's running in Hadoop. Halvade is written in Java. What Halvade does is, first of all, it provides a tool for uploading and partitioning your data. It manages the parallel threads, right? So it manages the parallel data. It converts your FastQ file to key value pairs using Hadoop BAM. It invokes your BWA instances, so either BWA align with SAMP or BWA MEM has support for that. Then it converts these SAM records to key value pairs, right, which are being sorted. And then you have all of the data preprocessing steps and all of the variant calling steps. So it's not as easy as just running GATK, right? Because you have to produce BET files. So we delineate our genomic regions using BET files. It has tools for automatically determining the optimal number of instances that you may want to run on a certain node. And then in the end, there is another MapReduce job that aggregates the partial VCF files that come out of all the reducers from GATK, right? So again, using the Hadoop BAM to parse all of the input formats.
So I have one question for you, Jan. When I see these figures, I'm really impressed. I think, wow, this is incredible. You're getting incredible speed up. But how do bioinformaticians tend to react? Often I find some of them might get defensive rather than be just basically impressed. I'm going to have this in my pipeline.
No, I haven't had this experience. Well, the paper is still under revision, so it looks good. So I think it will be published hopefully soon now. We will see what is the reaction of bioinformaticians when this happens. This is the second time we present this work in public. The first time was during the Genome Informatics conference in Cambridge in September, and I think we got very positive feedback. So at least not that I'm aware of any negative feedback.
Great. Okay, thanks again.
Okay, thank you.